Everyone, and welcome to this month's Insights and Practice webinar from Microbial Insights. Today, we're joined by Dr. Paul Erickson and Jack Sheldon for this presentation titled Evidence-Based Rapid CVOC Remediation with Proof, Linking Microbial, Geochemical, and Analytical Data. Dr. Paul Erickson is the Vice President of Research and Development at REGENESIS Bioremediation, where he oversees the commercialization of new environmental solutions and products, provides technical guidance on complex remediation projects, and participates in numerous research programs. Jack Sheldon is a senior consultant with Antea Group, where he advises on remediation technology selection, performance, and optimization on sites across the US and abroad. In addition to his decades of industry experience, Jack is also a subject matter expert on PFAS, other emerging contaminants, and their risk management. Today, we're talking about conducting multiple remediation strategies successfully and demonstrating that success with multiple lines of evidence. Our speakers today have worked with us here at MI for many years, so I'm super excited to continue learning from their expertise and seeing the application of our tools as one facet of complex site management. Before I hand this over to our speakers, I do just have a couple of reminders. First, at the end of this presentation, we will take our remaining time for a Q&A session. So please type your questions into the GoToWebinar control panel, and we'll get to as many of those as we can. This session is being recorded, and the recording will be available on our website as well as our YouTube channel in the coming days. You can also subscribe to our YouTube channel to help keep up with future webinars as well as our MI video series. Finally, certificates of attendance will be automatically emailed tomorrow to all attendees on the stream right now. Now, please join me in welcoming our speakers, Dr. Paul Erickson and Jack Sheldon.
Thanks, Hannah, and hello, everyone. Glad you could join us today. As you can see from the title and what Hannah mentioned, the theme that we're going to talk about is chlorinated solvent remediation and some of the tools that we use to do that, and the data that we can gather to demonstrate the proof, through different means, different lines of evidence that not only is the performance that we're after or that we're getting at, that the underlying mechanisms are there, that we're able to see what we want, which is full destruction in the case of these chlorinated solvents. So let's jump in. First, a quick agenda. So I'll introduce the concept of enhanced reductive dechlorination, kind of set the stage with some of the tools that we're going to use to remediate chlorinated solvents in situ, which is what we do here at REGENESIS. And I'll walk through some of the performance data that a pilot that I'll talk about, some of the tools that we use to assess performance, demonstrate through multiple lines of evidence that these contaminants that we're trying to treat, that we're taking them fully through onto end members that we're not concerned about. Show you that in this case in the pilot study that I have before I hand things over to Jack for a case study, and he'll wrap things up with some closing remarks. So a refresher for many of us, and hopefully a little new information for others. So enhanced reductive dechlorination, or ERD, it's a bioremediation strategy that's useful for chlorinated contaminants, where certain microbes can use these chlorinated solvents as basically something they can respire onto. And so their energy is coming from something else. In this case, it's going to be hydrogen. So compounds like PCE, TCE can be respired onto by certain bacteria, which you can see here. So Dehalobacter. The big one that we'll talk about mostly today is Helicocoides, and then generate these successively less chlorinated species, passing through vinyl chloride, which is toxic. So we don't want to spend much time here, but ultimately generating ethene, which is a gas that is innocent. We're not concerned about. And this last step in particular.
Paul, I'm sorry to interrupt you, but I think your screen is frozen. We're not seeing the slides advance or your presenter view right now. It's kind of frozen at the moment.
That's not ideal. What about now?
I'm still seeing the same.
Has anything
Yep,
changed?
now it just advanced, but we're still seeing the, we're not seeing the full view of the screen. We're seeing the.
How about now?
Nope, not on my side.
All right. Sorry, folks.
Yeah, sorry about that.
But you can see this part, right?
Yep, we can see it. It's fine. Yeah, if you collapse that, that will work great.
Okay, let's
We
go
can
with
keep going.
this. All right.
Perfect. That's
So
all.
I
Thank
already
you.
spoke through, yep, no problem. I spoke through the gist of this. So, really what I'll jump to is that we're looking to, in order to push this process through, we want to generate hydrogen and keep the bacteria that we either add that are present, the D. helicocoides, these dechlorinators, keep them satisfied and they'll keep things moving in the direction we want, which is the turnover of our chlorinated solvents onto end products here. So we can combine this process with sorbent. So in this case, colloidal activated carbon. Here at REGENESIS, we have a product called Plume Stop, and we can co-inject ERD using Aquafix, which I'll mention later. And this allows you to both support that process that we were looking at, this ERD, and get rapid removal first of your chlorinated solvents from solution. And this is a way that you can control your mass flux, get immediate drops in concentration, and get this biodegradation process to occur within a smaller footprint. And so the way that it looks in practice is you can get desorption, you get colonization of this activated carbon, which is where your contaminants have absorbed out onto. Biodegradation can follow, which then frees up absorption sites and then allows this process to continue. And so with this, you're able to get long-term chlorinated solvent treatment, CVOC treatment, through both the absorption process. You can also couple it with abiotic degradation, which Jack will touch on in his case study. But really what we'd like to see is evidence of this. So we can see absorption, and everyone sort of understands that. The compounds get removed from solution, but then really what we want is evidence that this biodegradation step is happening. And so that's what we're going to talk about today, is the data that we collected to demonstrate this. And in this context, in the field rather, using both Aquafix, which is this donor that doesn't interfere with Plume Stop that we developed, and see this in practice. So the evaluation that we're going to do is based on a number of factors. So most obvious is our solvents, our chlorinated solvents. We're going to be monitoring for those. Dissolved gases, methane and ethene in particular. Methane tells us that the conditions are right for this dechlorination process, because being anaerobic, you want to be mildly methanogenic in order to be under the prime conditions. And then ethene, that's our final end product, so we want to be looking for that. TOC is going to tell us that there's enough organic carbon that the fermenters can use to then generate the hydrogen that the dechlorinators need, so we're going to keep an eye on that. And then one big line of evidence is this microbial population that we collected these data. This is qPCR, which is available through means that Microbial Insights offers, like this quantarray. And that's going to tell us, again, are the right microbes present, these dechlorinators, and are they in appropriate numbers to sustain the process that we're trying to drive forward. So bringing it back to that slide that I showed before, on the microbial end of our analysis, we're going to be looking at a number of genes, and both functional genes and identity genes. I'll call this 16RS, but I'll focus my data on these two particular genes, so the DHC and the VCR. You can think of this first gene, the DHC, tells us really the presence of D. helicocoides, so it's just an identity of that particular species of bacteria. And then the VCR is the hardware that this bacteria can have. It doesn't have to have it. The helicocoides being there could be enough, but this VCR gene is, this is the gene section that codes for the proteins that are responsible for this final step of taking cis DCE and vinyl chloride and pushing them on through to our final end member. So we're going to look at data for both of these genes. Some details about the layout for this pilot test. So we did two different areas that I'll talk about today. One, we just used our donor, and then I'll follow that up with where we use both the donor and our activated carbon product Plume Stop. And just some quick information here about this. This was done in partnership with Hamp and Matthews on a site in Michigan. And the layout you can see here was we put our injection points in front of the well that we were doing our monitoring. groundwater was flowing from these injection points here through to the well. So let's look at the data. And in this case, again, just using the donor, you see that you get pretty rapid, within 3 months, conversion of our TCE, which was all that was present initially, onto cis, as we saw in our diagram, then onto vinyl, and then finally ethene. And you can see that the TOC, our total organic carbon, sort of plateaus here over the 2 years that we were monitoring and stays kind of in a nice sweet spot that is able to drive this dechlorination that we're after. And our genes that we're after on the microbial side tell the same story. So low concentrations, near non-detect in the beginning, and then they jump up and they're sustained kind of where you want. Typically for dihelicocoides, 10 to the 4 cells per mil is really your target. That or above is considered useful for biodegradation, this dechlorination process to be driven forward. So when we combine that with plume stop, this is what the pilot test looked like again. This is a co-injection of both our Aquafix and the plume stop. And again, groundwater is moving in the direction past the injection points to our monitoring point. And again, in this case, we had much higher concentrations of our ethenes. And you can see that here with our TCE and our cis. Combined, we were looking at over 40 milligrams per liter initially of TCE and cis. So this first rapid drop in concentration, that's really coming from the absorption of our chlorinated solvents onto the activated carbon. But then later on, you see that not only does the concentration of these chlorinated solvents not rebound, but you're seeing some of the ethene, which sort of glazed over in the first case, but you're seeing ethene show up here, which that's really what we want to see. That's the evidence that you're getting full mineralization of these chlorinated solvents within this activated carbon donor barrier. We're pushing everything onto end products, which is ultimately what we want to see. And again, our TOC over those 2 years drops, but then sort of levels out kind of where we want it to be. And what I'll do is I'll show this data in a slightly different form that kind of drives one of the points home, which is that we've got initially, if you look at this black line, which is total molar concentration of all of our all of our ethenes rather, you can see that there's a drop from that sorption again, but then what's present in solution, it sort of levels off and you don't get full mass balance, because what this on a molar basis, this black line, if you were accounting for all of the mass that's in the system, your moles would be consistent. All the moles of TC and cis are being converted stoichiometrically over to ethane. So that would be a flat black line across the top. So we don't have a perfect black line, but you can see that you get quite good conversion from our chlorinated starting products off to ethene. And again, you can see that in the molar fraction change here, where it's in the end dominated by ethene, which is exactly what we wanted to see. And again, as we saw in the case where we had the donor by itself, we've got the gene counts, which were initially low for both dihelicocoides and that vinyl chloride reductase. They've sort of leveled off into a useful spot long term. And then last bit of data that I'll show before I close and then hand things over to Jack was we had taken some of these cores from the site a couple of years ago and sent them to some collaborators of ours, Bertha Kellerup and her grad student Amir Riahi, who's now at I think Hazen and Sawyer, and they took some images, both SEM images and then confocal laser scanning microscopy to show. And again, these are images from the core samples that were collected from the site within the area that we had both no plume stop present and also with plume stop present. And you can see that the SEM images show the roughness going up here, and you can really tell that these are the activated carbon particles sort of speckled across the surface of our sand grains. And what's really interesting is that you can also see that this isn't the same image, but in through the CLSM imaging, you can see co-location of the bacteria, which are tagged here with the DNA is tagged with a fluorescent green marker, and you can see that they're co-located along with the colloidal carbon. So we're seeing evidence of our products, our chlorinated solvents generating these end members, ethene, which is what we want. We're seeing that the bacteria genes that are driving this process, they're present in the water. And here's some imaging data to show that in the field even you get this co-location of these bacteria that are responsible for driving these processes in and around these activated carbon particles on the soil. And so one kind of final thought on that is this study that they did there at the University of Maryland, they've got data from the lab that also sort of from an imaging standpoint look very similar to what we were able to pull from the field, which is exciting. So the takeaways on from my bit before I hand things over to Jack are that you can combine these absorptive technologies with something that like Aquafix, which helps drive enhanced reductive dechlorination, and this can be a highly effective flux management strategy. And I hope I showed you briefly here that through these multiple lines of evidence that we're able to collect, you can really demonstrate we're past this time when all we had was the ability to just look at our chlorinated solvents, see that they're going away from solution, but now we're able to show that even after they've been absorbed in these systems, they're available for biodegradation. And we've got the evidence from the genes for the bacteria, the visual presence of them, and of course that ethene final product is showing up as well. And so what I'll hand things over to Jack to talk about is you can also combine, when needed, these biotic processes with abiotic processes such as ZVI. And with that, I will pass things over to Jack, who will talk about his site and hopefully have better luck with running the slideshow here.
I mean, you may have to run the slides from your end.
I can hear you, Jack. Can everyone else hear Jack now?
Yes, we can hear him, Paul.
All right. So Jack,
Can you put the slide deck back up? It froze on my end, so perhaps we can put it back up and I'll just cue the slide movement.
Yeah.
I've made you the presenter again, so you should be able to share your slides for Jack.
Gotcha. Let's do that. I will try. I'm going to try screen this time, and let's see if that works better.
All right. Thanks, Paul. Since we're talking about technology, we had a little technology glitch there, but we're up and running. So building on what Paul just spoke about and the efficacy of this mixed multi-mechanism approach to remediation, I'm going to talk you through a quick case study as we wrap up today. And this is an exciting project because it's a large-scale application of this mixed multi-mechanism amendment. And what you see on the screen there to the left is this large site that had numerous buildings that were being demolished. This was a site that was undergoing a rapid-paced real estate development. And the three yellow areas that you see, the one toward the top of the screen that has the arrow pointed to it, that's the area we're going to talk about today. The other two areas were also remediation locations on the site. We're going to talk about that top rectangle that's 600 feet long by 330 feet wide. And within that, if you look to the right on the slide, there's a small pilot test area that we went out and we did a little proof of concept work around MW28 for this site. Paul, next slide, please. So the site scenario, this is a former oil field chemical site in the southwestern US. As I mentioned earlier, the developer was right on our heels as we were remediating. The site had changed hands from a property transaction standpoint. and they were not wasting time. They were tearing down buildings and ripping up foundations and really stressing the health and safety aspect of the project to the maximum. We were able to get out there and do a good amount of MIP work to help target our remediation area. We combined that with lots of conventional sampling of soil and groundwater. And of those 3 remediation areas, we used ISCO initially in each of those areas, combined with a bit of excavation and then backfilling with ISCO with oxidant within the excavations to give us a little extra treatment. So that was the starting point at each location. But then we got into the component where we built out this multi-mechanism amendment. So next slide, please, Paul. The area that we're treating in is characterized by a mix of silts and clays with some sand seams built within it. If you think about the figure I showed you at the very first slide, groundwater basically moves from right to left with some slight deviation to that. We're talking about chlorinated solvents, as Paul did in his part of the presentation. And we're mostly talking about TCE and daughter products, although we do have some 1,1-DCA and 1,1-DCE at this site as well. What made this site extra challenging with this new amendment is that we had a vertical component of upward pressure, almost artesian, that forced groundwater up in the zone. So we were constantly contending with that. And that made for some challenges as we were out doing the injection. Next slide, please. So this whole concept of a remediation toolbox, and we as remediation practitioners are always faced with the challenge of which technology do I select for my particular issue on my site? Well, isn't it nice when you can select multiple technologies, blend them all together in one tank, put them all in the ground at the same time, and achieve the various treatment mechanisms that Paul described so well earlier. So that's the whole basis of the approach here and where we went with this large site. Next slide, please. We did a pilot study on this site. It's always good to work the kinks out a little bit. We wanted to understand how best to mix these amendments together out in the field. We wanted to see how we could get them in the ground based on this strong upward vertical component of the groundwater. And we always want to collect lots of data going in, not just analytical, but field parameters and geochemistry. So we set a baseline for where we're going down the road. We did quantarray chlor with Microbial Insights, and we did have a hardy microbial population present, lots of dechlorinators with active gene functions in place. But we also use flux tracers in the pilot test well, and then we ultimately use them in full scale to look at how groundwater was moving in specific vertical intervals, as well as look at the mass flux in specific vertical intervals. And this proved to be a really valuable tool in terms of dialing in our amendment dosage. Next slide, please. So microbial data showed we had sort of an average population of microbes present. Again, we did have dechlorinators. The site had seen chlorinated solvents for decades. And so it was not surprising. We had a wide selection of dechlorinators present. And we had some activity in terms of gene functions that were present as well. So our goal here was to look to see if we could increase these numbers, these concentrations of microbes and enhance gene functions, and do so not only in the pilot study here, but then ultimately carry this over into the full scale on the project. Next slide, please. Here's a quick look at the flux tracer that was put in place for 2 weeks at the MW28 location. And if you look here, you can really see that about 16 feet below ground surface to about 18 feet below ground surface, we start to see more mass flux. We start to see a bit more movement in terms of groundwater. And again, this is so useful in terms of dialing in amendments and getting your most amendments where you need it most. So we do this pretty regularly on our sites now, and I think it's saved clients thousands and thousands of dollars because amendment is put where it's actually needed. Next slide, please. So in our case, we did add on the sulfadated S-micro ZVI product from REGENESIS to go along with Plume Stop and Aquafix and BDI. So the plume stop, again, for the sorption component, the S-micro ZVI for an abiotic reduction function, the Aquafix for long-lasting, sustained electron donor, and then the BDI culture to give us a little extra juice from a microbial standpoint. And again, so the first time these constituents could be mixed together. And that was the real beauty of this application. To inject these separately would have cost considerable amount. And it's nice to have amendments that are compatible, that can be readily mixed and put in the ground. Next slide, please. We used a bottom-up injection technique for the pilot study here. That is the actual pilot study area with all the commotion going on around it. We used a 2 to 3 foot long injection screen, about a 1.5 diameter injection tool. And we laid out a small grid of 3 rows of 4 points with 8 foot spacing between points, 5 foot spacing, or 5 foot spacing between points, 8 foot spacing between wells. And this was all done through direct push injection. So next slide, please. So some things that we observed, we generally had a very low flow rate from about 2 to 5 GPM. The average was about 2.4 gallons per minute. And we were able to sustain fairly low injection pressures at 20 PSI, which is ideal. We did have, in some locations, some surfacing. And as a result of that, we had to balance out the injection volume at some of the other points. We had some mixing challenges, and I would just put this out as a caution for any injection project where you're injecting amendments. Don't shortcut the mixing time. It's very critical when you're using amendments that you mix those in fully. And if it takes 30 minutes instead of 20 minutes to mix a batch, well, so be it. You need those materials to solubilize in order to inject them effectively. And we got a little bit of clumping. That was an observation during the pilot test. And with 10 extra minutes of mixing, we were able to get a nice smooth solution that we could put in the ground. We did find in the injection or in the monitoring well, in addition to a piezometer that we installed within the pilot test area, a black material. A little bit of the ZVI and the plume stop had found their way into those locations. So we did a flushing exercise with water to drive those materials further out into the formation. So those are some things that we learned during the pilot study. Next slide, please. This is a look at the layout. The MW28 monitoring well is right smack in the middle. The piezometer was just in that down gradient edge of the pilot study area. And we were able to monitor from both of those locations. So that's what it looked like at the pilot scale level. Next slide, please, Paul. A little bit on the setup out on site. This is the injection rig right near the monitoring well where we did the pilot study. We have the injection trailer with the mixed tank and makeup water. This site did not have any utilities available to us, so water had to be trucked from a fire hydrant, which was a half a mile down the road. So it made for some logistical challenges there as well. But these are all the things that you have to think about when you're out on a large remote site and need to put these things into play to conduct your injection process. So next slide, please, Paul. So what we see, we monitored at 30, 60, and 180 days. We saw a very rapid decline within that 30 to 60 day period where we would see concentrations go down. And we attribute this, much like Paul described, to sorption and a likely reaction with the zero valent iron that was present. Meanwhile, we were conditioning for the bioremediation component of this. We saw DO spike a little bit as we were injecting and then rapidly decline. We saw our ORP plummet. By the 180-day time frame, we had seen our ORP drop to minus 400 millivolts. And then we had evidence of reduced species in our geochemistry. And our microbial profile that we tracked in MW28 and the piezometer, we were able to show at least a 2 order of magnitude increase in our dechlorinating bacteria and increased in concentrations in our gene functions. So it's exactly what we wanted to see to have the confidence to take this methodology, this multi-amendment mechanism forward to full scale. Next slide, please. This is just a quick look at some of the concentrations that we ultimately achieved at the end of the pilot study. The injection event began right in the middle there in August of 2023. We wrapped it up in March of 2024 with our last sampling event. And you can see we had significant reductions in pretty much every constituent that was there. And those were the lowest concentrations that well had ever seen historically. And we'd been analyzing samples from that well for almost 10 years. And those were the lowest concentrations achieved. So this gave us great confidence moving forward that we understood the mechanism that was going to be in play, as well as the potential that this technology could lead us to on a site at full scale. So next slide, please. It's just a graphic view of that again, and you can see we've had orders of magnitude decreases in certain constituents. It was always good to see vinyl chloride also decrease, and somebody will ask the question, and I'll answer it now. Did we see ethene and ethane? Didn't see ethane, but we did see some ethene pop up in the well as we got to the latter stages of the pilot study. And when we see that, we're always encouraged once again that we're getting full dechlorination. So next slide, please. So ultimately, armed with that information from the pilot study, we were able to take this to full scale. And the way we did that was a series of barriers along that large plume. And these barriers are staged up gradient of monitoring wells. So we had an ideal monitoring well network. So we'd have a barrier, a monitoring well, a barrier, a monitoring well, and they were spread out across the length of that plume. And we were able to play around with the design here to limit the width of our barriers to really just capture those core zones we needed to treat. We used the flux tracers to help dial us in on the vertical component. And for each of those barriers, we had a different formulation. So we had a different dosage, essentially. So we're able to dial in different aspects of the aquifix, the plume stop, the BDI, the zero valent iron. So the recipe was customized for each barrier as we went. You'll notice that most of the barriers are pushed back toward the original source area, which is the right side of the screen. And these were staggered really nicely. There were perhaps 20 to 50 feet apart at a maximum. And then we had two down gradient barriers toward the property line because we had seen some low concentrations starting to emerge at wells down there. So this was a migration control approach, but it was also a heavy duty barrier application to treat any residuals from the source that were making their way down. Slide, please, Paul. And 400 injection points later, done over a period of about 2 months. And these are the overall results averaged out for the monitoring well network. And you can see that in a very rapid period of time, we did the pilot work. And then ultimately, we saw in the full scale rapid decline and very sustained concentrations over time. Right now, this site is at a point where most of the monitoring wells are below standard for all the constituents. We're just dealing with some minor concentrations of vinyl chloride at a couple of locations. We did achieve enough degradation with the barriers, enough treatment with the barriers that the down gradient wells near the property line no longer have concerning concentrations. So this particular approach was very successful because of the multi-mechanism amendments that were put in place. And let's go to the next slide, Paul. This is MW28 at full scale. Again, we've crept down with several of our constituents since the pilot test. So we continue to see those concentrations decline. And then next slide, please, Paul. MW19 and 19A was further up toward our source area. And you could see there too, in many cases with the constituents, we've had at least an order of magnitude decrease. So next slide, please, Paul. So what did we learn? We did learn that we could do direct push injection into these artesian type conditions or the strong upward vertical component. And the way we did that was we staggered injection points within the barriers, and then we went back to locations. We balanced out a little bit of the volumes from point to point, and we used basically a capping technique to push the pressure down at individual direct injection locations. And that methodology overall helped us keep amendment in the ground and limited surfacing. We learned, again in the full scale, that we couldn't skimp on the mixing time for this multi-mechanism amendment series. We needed to spend the time mixing the batches, and on average, that was about a half an hour to mix a batch in order to be ready for the next series of injections. We did learn that sorption and reduction occurred within the first 60 days, and that acclimation for bioremediation took place over about a 90-day period. We started to see daughter production then, and then those daughter products started to decline. So we saw a nice steady treatment regime with all these mechanisms working in concert together. We learned that mass flux and microbial tools are absolutely essential to projects like this. It gives us extra evidence, extra parameters that we can track. I don't do a single project anymore where we don't try to get mass flux built into it or microbial profiling built into the project, because it's that extra layer of data that builds out your conceptual site model, and it's something you can track as the project goes on. This was the first full-scale application of this amendment. Paul had done the first pilot study. Our pilot study was right on the heels of that, but this was the largest and first one of this kind. And the barrier type location now, our approach is one that we've duplicated from site to site with very similar results. So this combination of amendments is a winner. It gives you all those features that you're looking for, speed and thoroughness of treatment. And I think we've got a real sustainable type product here. We can have long-term electron donor release. You can have flourishing microbial populations. You can have sorption in place and reductive materials in place for a long period of time, which will help you manage mass flux and diffusion, back diffusion on these sites as they go forward. And next slide, please, Paul. And with that, I think we got done in a reasonable period of time here, and we're open to questions. Thanks for being the projectionist, Paul.
No problem.
Did a great job, Paul. Thank you very much, Jack, too. The presentation was amazing. Paul, you did such a good job. It's really impressive to see how much we've learned about these technologies and good application practices for what we can do in the field. So it's exciting to hear both of you guys speak today. We do have quite a few questions. If the audience has additional questions, please enter them into your GoToWebinar panel. We do have a little bit of time here to be able to get through these. The first question is for you, Paul. How do you see differences in the absorption and release of the TCE and the daughter products on the plume stop? Do you see differences in those, and how do they compare with different TCE or different daughter products?
So in a barrier situation, if things are working properly, you rarely see much daughter product showing up, and really that's because there's enough capacity in the system so that you can get full degradation as the components are sort of moving with the groundwater and along the treatment area. With that said, if you've got enough transformation products that aren't being converted, those elements, so going from TCE to cis, for example, and cis to vinyl, they do sorb less, and so you would certainly see them migrate faster and potentially show up in the dissolve phase sooner if you don't have this sustained degradation that's driving them on towards ethene going on.
And I think this is a good follow-up to this, Paul, and maybe Jack might have some additional thoughts on this based on field implementation. But another question is, are you seeing any reduced permeability with the barriers over time, or movement of groundwater that happens around the barriers, and kind of how would you deal with that?
So we do take care to keep our dosing sort of within the ranges that you're going to minimize the changes in permeability, because you are indeed putting in solids. These are colloidal solids, but they're solids nonetheless. It turns out that you're only using, the doses on the carbon basis. For these chlorinated solvent jobs especially, something like 1,000, 1,500 milligrams of activated carbon per kilogram of soil. That amounts to something, a couple of percent of the pore space is then being filled by solids. The other amendments add to that, but we've done lab studies to show that the permeability changes are on the order of a few percent, and manageable with planning about how you extend the edges of your barriers to make sure that you don't have short circuiting. And then with the donor that we use in particular, you're not getting overwhelming amounts of biofilm formation, because the carbon release is you're aiming for a minimal amount of carbon release with time. So you can have the same issues with EVO products. It is something that has been known in the industry for some time you have to manage. I don't know, Jack, if you have anything else that you want to add to it.
Yeah, I think that sustained slow release carbon, Paul, is a critical feature here. And we've not seen any permeability issues. We've done this now multiple times on sites, and it looks very much the same from site to site. And we've done lots of work with plume stop and all sorts of bioamendments of different viscosities. And yeah, realistically, because this is slow release in nature, we just don't see any impact on permeability at all.
Great. All right, Jack, this is a question for you. With the combination of the amendments that you were using in one of the site examples, have you seen any issues with methane generation or volatility of the product during the injections?
Not so much volatility of the product. I will say we have seen some differences in methane from site to site. We didn't see it at this particular site that I talked about today, but other sites that we've done since that time, we've seen a little bit more methane production. So I think that's obviously a function of the microbial makeup at the site, which obviously can look different from site to site. But that's something that we always track. We try to understand methanogenic conditions at sites, and it does look like it varies from site to site.
Yeah, I think it's important to understand methanotrophs and methanogens interaction so you can see how quickly that methane will be utilized if it's formed in situ. So that's something the quant-array analysis that Jack showed you could help you to understand how quickly that will be transformed.
We have seen that plenty of times on ERD sites where even when you get methane production at significant levels, if you've got a robust methanotroph population there and a little bit of oxygen, off you go. There goes the methane.
Yep. One of the questions, Jack, is they missed the total size area that you used over those 400 injection points.
600 feet long by 300 feet wide.
Awesome. And then you mentioned information on flux tracer. Are there other tools similar to, like, are you talking about like iFlux, or can you discuss that a little bit more?
There are other tools in the marketplace that give you Darcy velocity and mass flux. We've gotten accustomed to the flux tracers now for chlorinated sites and for PFAS, but there are products available from EnviroFlux for petroleum applications. We've seen iFlux come over from Europe, and that has some interesting features associated with it. So the short answer is there are other tools, and they can be very specific for different applications.
When you were looking at the radius of influence, you were talking about 2.5 feet, and that for some sites could be small. Is that due to the lithology of the site? And can you talk about that radius of influence and what you saw in the pilot study compared to like a full scale application?
I know we were able to increase it for the full scale application. We want to be very cautious because we did have a fair amount of clay and silts at this particular site. Even though we had some sand seams, we were able to find our chlorinated solvents often in those sand seams, but we still, we still had those considerations for sure. The site was very challenging with the artesian conditions. I'll call them artesian. Maybe you can debate whether they were or whether they weren't. But the vertical component gave us lots and lots of challenges. So as a result of that, we really had to work that scenario. We were very conservative when we did the pilot study, and then we learned from that, and we were able to build out the full scale.
Can you talk about that vertical component a little bit? There's some questions about the depth that you were treating.
Yeah, we ultimately went deeper than even just our flux tracer told us, to give us a little conservative approach in some areas. In some of the areas, the flux tracer did give us a wider or a lengthier vertical component. So we injected from 9 to 21 feet below ground surface. I think the pilot study one I showed, we were like 16 to 18 was where the majority of the mass flux and the movement was, but it varied from location to location. So we use that information then and build it into the design. And I think the average injection vertical interval was 9 to 21 feet with some deviation from barrier to barrier.
Great. One of the questions, Jack, is that someone's considering using this at another site in North Carolina, and they're talking about the molecular tools. And so if you did the quantarray chlor in this instance, are there other technologies or tools that you've used with MI that you think would be beneficial? And I'm also happy to add some context to it after you finish as well.
Yeah, I think there's such a wide array of tools between next generation sequence. It depends on the level of microbial information that you want to obtain. We really like the quantarray because it gives us a look at all these different potential mechanisms at a given location. We spent a little less money and done census qPCR from time to time. And then we've mixed up our microbial tools from site to site. But we've tended to rely on the quantarray because it gives us such a broad snapshot of what's going on at a particular site. And I don't know if you want to supplement that, Dora, with some other things. But I mean, we've used not on chlorinated sites, but stable isotope probing in different places. And we try to use as many of the microbial tools as we possibly can. And now your selection has broadened over time. So if you want to fill in the blanks there a little bit.
Yeah.
I can also just,
sure.
Before you do, Dora, we always use microbial tools on our beta projects and pilots when we're testing new technologies, because
Yep.
we sometimes get surprised by what we see, sometimes in good ways, and other times just things to watch out for. So there's enough rich information, both on the anaerobic and aerobic side, even when you're looking at a quantarray chlor, that we find it an invaluable tool to use on the development phase of our technologies.
Absolutely.
Awesome. That's good to hear. And I think for if you're talking about a chlorinated site, quantarray chlor is meant to encompass your anaerobic, co-metabolic, direct metabolism pathways. So you kind of get that whole package there. But if your site is mixed and you have other contaminants, there may be other qPCR assays that would be beneficial to you. Or let's say you have abiotic influences that could be happening at these sites. We have a quantarray for biogeochemical that could help you understand iron sulfide formations and iron cycling and nitrogen cycling. So kind of depends on other things that are going on at the site, or if you have concerns about other receptors or beneficial reuse of the land, understanding nitrogen cycling could be beneficial to you. So there are some things that could complement it. So if you want to reach out to us and give us specifics on what co-contaminants may be there or what challenges that you're experiencing, we can let you know if there's others. But for the vast majority of chlorinated projects, quantarray chlor is designed to be kind of all-encompassing of the known pathways that are out there to give you information to make good decisions. Like Paul and Jack were saying, I think it gives you kind of the breadth of what's going on at your site assessment, so you can make those decisions, or know, this pathway is not likely dominant, so I'm going to have to do a lot of work to make this to occur, or I may have a limiting factor there that I need to consider. And, another question that complements this, Jack, is someone asked about bioaugmentation and if you had any concerns of needing to bioaugment at this site. And so I just wanted to see kind of what you thought about that, and if you are using kind of baseline microbial data to help you understand if you can biostimulate, and kind of what your rules of thumb are for that type of situation.
Yeah, I think we definitely look at a baseline and then make a determination. If we figure that our microbial population could use a little supplementing in order to really be at peak performance, then we take that step. I think in this case, as much as anything, we were really interested in the combination of amendments. So it was going to be in this site if we could possibly build it in. And so that's what we did with it. But that's usually where we start, with a baseline, make a determination of who's there and in what concentration, what are they doing, what are we trying to achieve, and how does it look across the site? One portion of the site relatively anemic, as opposed to other portions of the site where the microbial populations are flourishing. So all those things come into consideration as we decide whether or not to bio-augment.
There's actually a few different questions, Jack, that relate to kind of the design phase of how you came up with what you were going to do here. With, you ended up with 400 injection points, and you had, multiple barriers that you're setting up, trying to figure this out. How do you go about kind of coming up with this conceptual design and the extent of the barriers? Are there certain calculations or data that you would recommend that people kind of highly take into account in the beginning to help them do this, the most economical and straightforward path? There's kind of several questions that lead to that same conclusion.
There's always design calcs, looking at dosage and movement of injection fluids across the area. So that helps us with spacing. It helps us with spacing rows in this case for barriers. So that becomes a critical feature. I didn't get into it in the case study today. We probably could have added an extra slide, but a conceptual site model was built out, and we used that as a tool with a lot of robust data built into it to look at the whole remediation application and what that could look like. And then ultimately, when we get data, performance data for a remediation project like this, can add those data into the model and see the plume change, hopefully, and use that as a performance monitoring tool as well. So there's a lot that goes into it. We take, the microbial piece, we take the flux tracer piece, all those things in concert. We work back and forth with the remediation vendor company, in this case, REGENESIS, and that's how we build out our design.
And then in this case, you had some groundwater concentrations that were pretty high. Did you have concerns about source material that may be remaining there that you needed to address, and how did you take that into account?
Yeah, we knew that we would need multiple barriers out here because we had to get in and get out of the original source area really quickly. I can't understate how challenged we were on this site with the development activities that were going on. They were literally on our heels. So we had to do an excavation in the source area and quickly add ISCO or oxidant into the excavation, close it up, backfill it, be ready to move on from there, do some very limited injection of oxidant around that excavation area. And as a result of that, we knew that there would be some residual that would start finding its way down gradient. Hence the reason for all those barriers across that large area. So that became an important consideration.
Awesome. We'll do one final question and then kind of wrap up for today. And if people have other questions, I'm sure they can reach out to you guys directly. They've got your email addresses there, or you can reach out to us at info@microbe.com. But this one last question, I'd like to get both of your input, because it's something that I personally have run into as a challenge over the few decades that I've worked in this industry. And I think it's a struggle for all of us. But when you run into bedrock aquifers, you can get sloughing and biofilm formations and a lot of treatment variability and concerns that come along with bedrock. What special considerations do you use in the treatment of groundwater that are in these aquifers?
I'll try to take a first crack at it, and then Jack, please backfill where needed. We think much more clearly lately about flux and, where is the issue that you're managing going? And what tools can I use in order to put in place some sort of barrier that's going to prevent where the issue is from getting to where you don't want it to be. And when you try and deploy your amendments, your technologies in that way, it sort of gives you a plan in terms of where you can put them. Bedrock is always challenging, but we've had success with using methods where, when possible, you can you can flip your barrier, which typically we're thinking about putting them horizontally, but maybe put it vertically so that you're capturing mass that's migrating, you know, from above to below, if that's possible. So you can use these sorts of technologies. You just have to be creative and be thinking about flux and making planes of treatment, if you will, when and where possible. You know, it's easier said than done in Bedrock, but that's the way that we try and approach problems like this these days.
Yeah, I think Paul said that really well. I think that management of mass that's finding its way to fractured rock, I think that's a real key thought process. And the other thing that I would say is that in fractured rock projects I've worked on in the past, you can have different degrees of sediment that deposit within the fractures themselves. And that could mean a whole lot of mass in some of those fractures and need to really get in and contact those sediments and get treatment there. So you can have high variability within the fractures. I've seen some really cool projects too over time where if you can get that treatment aligned in the fracture in just the right manner and get it to transport a good distance through that fracture, you can get some surprising cleanup. But these fractured rock projects are always complicated. But I think that's a healthy thought process to really think in terms of managing what's making its way to the fractures. And if we can combat that, we can really lessen the impact of what migrates through the fracture network.
Yep.
Well, thank you both. I personally learned a lot during this presentation today, and I'm excited to see the advancements and the collaborations that we do together and have done together for many years. It's always a joy to hear you both speak. You're excellent presenters. And I know the audience really enjoyed your presentation today, so we're very grateful for your time and effort to put this together. And we appreciate everyone for attending and taking the time. If you have questions, please reach out to Paul or to Jack or to us at Microbial Insights. We are always happy to partner with you guys and to answer questions and work together as a team. It's something Jack and I were talking about yesterday. This is a team sport that we have to work together to accomplish the goals of cleaning up and doing all the work we're trying to do. So please feel free to reach out to us and stay connected. We will have more webinars coming up this year that we hope you'll join us on. And, please stay in touch and let us know if you have questions or thoughts for future webinars that would be of interest. And Jack and Paul, thank you both very much again for your time today. We really appreciate it.
Thanks for having us, and thanks everybody for joining.
Yeah, thank you.
All right. Thanks, everyone. Have a good day.
You too.
Bye.
Bye-bye.